General-purpose AI models have become remarkably proficient at predicting protein structures. But a question arises. What happens when your research hinges on a single, specific protein? Perhaps an outlier that the model doesn't generalize too well. A method called protein test time training, or ProteinTTT, has been developed to address this very issue. It's an approach that customizes a large model for a single target, on the fly, without the need for any new training data. In this analysis, we'll unpack how this method operates. We'll begin with the core generalization challenge these models face. Then, we'll examine ProteinTT itself, its underlying mechanism, the empirical data validating its performance, and its impact on real-world biological problems. Finally, we'll consider what this approach signifies for the future of computational biology. So, let's start with the problem that prompted this line of inquiry: the generalization challenge that is inherent to large-scale protein modeling. You see, modern machine learning models are trained to perform well on average across vast datasets that might contain millions of diverse protein sequences. An experimental biologist, however, is often not concerned with average performance. Their work may demand a high-precision prediction for one specific protein, and this protein could very well be an outlier that's poorly represented in the training data. This discrepancy between broad statistical performance and specific single-target accuracy creates a significant hurdle. This really sets up a fundamental conflict. The models are optimized for breadth, but specific scientific inquiry often requires depth. A model that performs well across 1 million proteins might still fail on the one that is critical for a particular experiment. This is precisely the gap that ProteinTTT is engineered to bridge. And that brings us to the proposed solution: protein test time training. It's a method that allows for self-supervised customization for an individual protein right at the moment of inference. The central hypothesis here is quite direct. The researchers posited that if a model could be adapted to better understand the unique sequence patterns of just one protein, it would then form a more accurate internal representation of that protein. A higher fidelity representation should, logically, lead to more accurate downstream predictions about its structure or function. Now, to quantify this level of comprehension, the method uses a metric called perplexity. In language models, you can think of perplexity as a measure of surprise. If a model accurately predicts the components of a sequence, it isn't surprised, and its perplexity is low. A lower perplexity score, therefore, signifies a better probabilistic fit, meaning the model finds the sequence's patterns to be predictable. This observed correlation is really the crux of the method. High perplexity suggests the model is struggling to predict the residues in the protein sequence. This poor fit then leads to a flawed internal representation, which ultimately results in lower quality predictions. The objective then is to minimize perplexity for the target sequence in order to improve prediction accuracy. So, how does ProteinTT computationally achieve this reduction in perplexity? Let's take a closer look at the specific mechanism for customizing a model at test time. Many of these models use what's known as a Y-shaped architecture. This consists of a large pre-trained backbone that extracts features from a sequence, a self-supervised head used for the initial training, and then a specialized head for the final prediction task. ProteinTT's intervention is quite targeted. It only adapts the large feature-extracting backbone. It achieves this by using the model's own self-supervised objective, in this case, masked language modeling, to refine that backbone, all while the final prediction head remains completely frozen. The customization workflow itself is very methodical. First, a single protein sequence is fed into the model. Second, the backbone is fine-tuned using the masked language modeling objective on that one sequence alone, with the specific goal of minimizing perplexity. During this optimization, multiple updated versions of the backbone are generated. A confidence function like the PLDDT score is then used to select the best-performing backbone. Finally, this newly customized backbone is paired with the original, frozen prediction head to generate the final improved result. Now, to make this whole process computationally feasible on models with billions of parameters, two key optimization techniques are used. Stochastic gradient descent is employed for its stable training dynamics, and, critically, a method called low-rank adaptation, or LoRA, is also applied. LoRA dramatically reduces the number of trainable parameters that need to be updated, which is what makes it possible to run this entire on-the-fly customization process efficiently on just a single GPU. With the mechanism now detailed, let's turn to the empirical evidence. We'll assess ProteinTTT's performance across 3 standard tasks in computational biology. The first area of validation is protein structure prediction, which is the process of determining the three-dimensional coordinates of a protein from its amino acid sequence. Looking at the data from the Cameo benchmark and focusing specifically on targets that base models predict with low confidence, a consistent pattern emerges. For 3 different structure prediction models, applying ProteinTTT improved both the global accuracy, measured by TM score, and the local structural quality, measured by LDDT. This consistent improvement across different model architectures points to the robustness of the method. This chart helps to visualize the performance gain for the ESM model on these challenging targets. The average TM score increases from 0.46 to over 0.50. That constitutes a notable improvement in structure prediction quality, especially for proteins that the original model found difficult to resolve. This second area of validation we'll examine is protein fitness prediction. This involves ranking the functional effects of different mutations to a given protein. On the ProteinGym benchmark, we observe a very similar pattern of consistent improvement. The average Spearman correlation, a metric that measures how accurately the model ranks the functional impact of mutations, is enhanced for all of the models tested. And when applied to the PRO-SES-T model, ProteinTTT actually establishes a new state-of-the-art performance on this benchmark. The third validation area is protein function prediction, which entails classifying a protein based on its biological role. And again, for 2 distinct function prediction tasks, classifying enzyme substrates and predicting a protein's location within a cell, ProteinTTT delivered measurable performance gains. Both the mean average precision and the accuracy improved, demonstrating that this per-protein customization is beneficial across a range of task types. So, having established the quantitative improvements on standard benchmarks, we can now look at the practical utility of ProteinTTT through 2 challenging, real-world biological case studies. The first case study addresses the difficult problem of predicting the structure of antibody binding regions. This is a critical task for therapeutic design. The primary difficulty in modeling antibodies lies in their complementarity determining regions, or CDRs. These are hypervariable loops that carry out the actual binding to targets. Because of their extreme structural flexibility, they are notoriously difficult to model accurately. On a set of challenging structures, ProteinTTT improved the local structure quality for 66% of these antibody CDRs and for 60% of their corresponding antigens. This provides a more precise model of this critical interaction. The second case study moves into improving the structural repository of viral proteins, which is an area of high importance for vaccine and antiviral drug development. The key finding from this analysis is a significant expansion of structural data for viruses. By applying ProteinTTT to the ESM model, the researchers were able to generate a higher confidence structure prediction for 19% of the proteins in the Big Fantastic Virus database, surpassing the quality of predictions from both the original AlphaFold 2 and ESM models. This chart helps to put that impact into context. The original database had high-quality predictions for about 55% of its entries. The base ESM model improved an additional 10% of the remainder. By adding ProteinTTT customization, that contribution was nearly doubled to 19%, substantially increasing the coverage of high-confidence structures in this viral database. And this brings us to perhaps the most significant insight from this work. ProteinTTT provides the greatest benefit for proteins with low MSA depth. In other words, proteins with very few known evolutionary relatives. This is critical because these are often the most novel or rapidly mutating proteins, like those from an emerging virus, and they are precisely where traditional, evolution-based models tend to struggle the most. All right, let's bring all of this together and summarize the key contributions of this research before we consider its future implications. In review, this work introduces ProteinTTT as the first test-time customization method in computational biology, and it establishes a clear link between its effectiveness and the minimization of perplexity. The method was validated empirically across structure, fitness, and function prediction. Its practical utility was then demonstrated in case studies involving antibody modeling and viral proteomics. Together, these contributions establish per-protein customization as an accessible and powerful tool for biological research. This leads us to a final and critical question. As foundational models in biology continue to grow in scale and power, the work on ProteinTTT points toward a new frontier. How else can we leverage this kind of on-the-fly adaptation to specialize these generalist models and apply them to the most specific and challenging problems that researchers are currently facing?